but I think as you obviously do, I think they're really important.
Absolutely, absolutely. Yeah. Great. So we are all one or the other way contributing toward Indian science, and I'm very happy that just ideas being able to link it to this whole meeting which is happening in Pratapgarh. We are not physically there, but I think we are able to communicate true science, which we all were involved, and also uplifting, sharing our knowledge and experience to this nation, which requires a lot to do. And I hope that such kind of trials would come up to really establish the things which have been proven by research, and a clinical trial can definitely be added to really bring out those informations and then make it a public at large. Yeah. So that is a good link further. Thank you so much, Lindsay.
My pleasure.
I would like to go ahead. What's up? Shall we continue now, or are there some more comments or questions? All right.
These two speakers are really wonderful, and they will bring some
We
model.
can't hear you guys.
Yeah.
We cannot hear you.
So you cannot hear me. Can you hear me now?
Okay, Harry, you can hear me though.
Yeah, I can hear you.
Good.
Okay.
I said two of you.
Right. So I would then. I think this is my turn, right? I'm not wrong.
Yeah, please.
But Musha, should I go now? I start now. Okay.
Please.
The people back in Pratapgarh, they can't hear.
I think just better to just make sure that they can listen to you.
Yeah.
We are listening. Is it all right? Are you listening?
Yes.
It's good.
Okay, shall we start? Are you guys hearing me?
Yes. Go ahead.
Yes, please go ahead, Harry. Yeah, we can hear you clearly.
So I think we all, those who are online, we all hearing each other. But I hope from them they are not hearing. I hope they are hearing us. I'm sure they must be hearing us.
Yeah, online it is very much audible, but I don't know what is the status in the live session.
Yeah, that's what I'm really surprised. But nevertheless, we will continue, okay? They would at least listen to us.
No, it's all right. We are hearing here. I think it's okay.
I can also hear you online.
So all the people online and they are hearing it, I'm sure our voice must be going there.
मैम आप कर दीजिए
I think they got muted, but nevertheless.
No, we are listening. We are listening here. I think your voice is not going there.
वॉइस इज
Okay, my slides are visible, all right?
Yeah.
Yes, it is visible.
Yes, they are visible.
We can hear you. You can start.
Okay, so
Yeah, they are very well visible.
yeah, okay. So without really wasting a minute online, because we all have to rush to our next commitments. I also have to check out here. So let me continue. Most of you think they know me, but I'll just say a few words because the Indian Academy of Biomedical Sciences, which collaborates or vice versa, the National Academy of Medical Sciences and other like ACBI now this time, for the first time we have done this session in ACBI on cardiovascular diseases. I'm very happy the other friends could join from different parts of the world. I would like to quickly take you through another piece of work which is purely done on the patients where we have identified certain genes very similar way what Kapil showed in his from bench to bedside study. I'm trying to do from bed to bench and then bringing the information back to the bed. So working on the molecular phenotype. In fact, this whole story starts almost three decades ago, when, as a young postdoc, I met Judah Fulpan, a pediatric cardiologist, I would say, from Children's Hospital in Boston, and very talented person who coined the term angiogenesis, because he was working in the field and I was working directly on an animal model back in Rotterdam, and we were identifying certain growth factors from the heart where I met him. So he is my inspirer. Unfortunately, he's no more, and the kind of role model, fantastic work which he has contributed in the field. So when we do research, particularly with translational research, there are four major pillars. Whatever we do, we have to keep it in mind from day one. Research and audit. So when we conduct the research, there is a continuous audit, means quality check, reference checks, the controls, negative controls, the positive controls, all sort of things, audits of that particular research. That would lead to the implementation of evidence, because by doing this research we generate evidence and we can implement. So very, very relevant aspect already in two pillars. But once this is established, the teaching is also taken further. We propagate what we have done, we publish, we teach to the students, and that's how the research is established, and obviously it creates the improved delivery of healthcare. So in translation science, these four pillars must be kept in mind all the time. I'll take you through, starting from the beginning, how we embarked upon the human heart failure. This is the work which we initiated in experimental animals, where we had the myocardial stunning protocol, where we did the putting a clamp in the LCX region and also measuring the systolic wall thickening in the non-ischemic area and the ischemic area by putting the ultrasonic crystals. This was done, all work was done in the large animals, pig. And you would be surprised, probably some of you, that pig heart is very similar to human heart in many, many ways. For example, collateral circulation. The human heart and pig heart, they have similar capillaries which are not really functional, but they can open up when there is a slowly opening ischemia. And that was one of the reasons we were working on the larger animals. So the protocol which we did to start with the stabilization period of occlusion and 30 minutes of reperfusion, and again 10 minutes of occlusion and 30 minutes of reperfusion. So doing these two episodes of ischemia and reperfusion, that results into many changes at a very acute level. They can persist for a long time during the reperfusion phase. So whatever is happening due to this ischemic insult, we would like to understand in this area during the reperfusion and for longer period. So any change at the mRNA or gene expression level will result into the proteins and then we can look at the functional changes in this heart. Then again I said that for the audits in every control, everything which we are doing is comparable, that the data which we are generating is absolutely relevant and directly linked to the work which we are, the protocol which we are using is the sham operated, where we did the thoracotomy and left, we exposed all the vessels, particularly the LCX region, and then left the normal reperfusion. We did not put the clamp here, but the rest were the same. So, and then different time points of reperfusion. As you can see in the hemodynamic changes which happens, systolic wall thickening, as soon as the ischemia, in the first episode there is a depressed wall thickening, we develop systolic wall thickening as compared to the non-ischemic zone. This is all measured using the ultrasonic crystals. And during the reperfusion of half an hour reperfusion, you see that this is returning back, but is still much depressed, significantly depressed. And in the second ischemic situation, then this again this hyperemic response is minimized, but it is taking long time to recover. So the ischemic tissue would take long time to come up to the normal function. But this adaptation phase is really the key for preparing this maybe the longer ischemic insult in the future. So this condition is called preconditioning, when this reperfusion stage has resulted into large number of molecules which are protective to a longer insult, and that one is called ischemic preconditioning, which was at one time was a very important event, and people thought that that could be helpful, but obviously in patients it's very difficult to do preconditioning. Rather, some of the molecules have been used, those we identified as a biomarkers, but there's still not much to learn before we can really embark upon these all, protocols when it comes to the human heart. So we wanted to quickly look what's happening. There are many papers from our group where we have looked at many components of cardiac function, the calcium regulation, the contractile proteins, the transcription factors like very, very early genes, the fibrotic factors, and all other kind of growth factors. But I'm just focusing on one right now because it's relevant to the human heart failure, the work which we carried on the patients. There we have looked at the BIOCHEMISTRY, the vascular endothelial growth factor, which is the key molecule for blood vessel formation or angiogenesis, which can happen in the heart. It has got two mRNA species of 3.9 and 1.7. And as you can see here, the myocardial ischemia, and during the reperfusion phase, there is a tremendous, the robust expression increases in the ischemic zone, which returns to the normal level, and we can compare that as a quantitative analysis. As you can see, there is a transient expression of BIOCHEMISTRY very early, and then it decreases. So this mRNA is a direct link to the, and this was the work when we were doing, we had no clue there was hypoxia-induced factor was not really discovered by then, receptors for BIOCHEMISTRY were not discovered. Now we know everything, how this is all happening, this whole mechanism we understand now very well, that the ischemia resulting into the lack of oxygen in the blood vessel, particularly in the cardiac tissue, resulting into the expression of BIOCHEMISTRY, and now it is obviously linked with the Hipron alpha, which was discovered a bit later, and we showed this data around 1990 and onward when we were working on this. In the long term, when you leave these animals, they develop collaterals, means that the ischemic zone is salvaged by linking and creating the network, opening up the collaterals, and the ischemic zone would be, having the supply of blood vessels, because this is this has been a very helpful situation as compared to the normal Sharma animals, which was all functional there, properly done. So this is all happening with the formation of blood vessel as well as arteriogenesis, opening up. So I'll take you through to the mechanism. Once we saw that in the animal model, let's get back to the humans and see where we see collaterals and how we can really help and increase the collateralization in the heart to help patients to survive. This is all in the event called angiogenesis, and there we are trying to look at the endothelial cells. It covers the whole blood vessel, and if you see that the total amount, and when we can dissect and measure the weights, something like 720 gram in an adult, about 600 gram capillaries. So very fine. Most of the capillaries in the whole body circulatory system, about 600 gram. And you would be surprised that they all cover about 3,000 square meter of the surface area. And if you put all those capillaries and larger vessels together, they will run to hundreds of thousands of kilometers. Incredible network in one human body. Very important, we need to keep them functional till we survive. So, to refresh your memory how the blood vessel looks like, as you see here, the endothelium, vascular smooth muscle cells, media, and intima. The building block starts with the endothelial cells. Here we can isolate and do a lot of other type of in vitro experiment to create models and all that in vitro. We can isolate all these cells very well and very specifically play all sort of different experiments now. So what exactly the mechanism by which angiogenesis occurs in the body, we understand well now. Starting from the hematopoietic stem cells, there is a circulatory system which brings all these cells in the contact to the stem cells, and platelets and CD8 and CD4 positive cells, they all secrete number of cytokines and growth factors. They all in a concerted fashion let these cells get matured into endothelial stem cells, endothelial cells, and that create a tubular structure. And this tubular structure will continue further to sprout, to grow, and create the vascular network. A lot of things happen here. Most of the rapid proliferation and all the actions are happening at the tip cell and the neighboring stalk cells, because those are the cells which are experiencing ischemia or hypoxia. And due to the hypoxia, a large number of molecules, especially the VEGF-A, 165 amino acid protein is the most relevant, which is direct nitrogen for endothelial cells to proliferate and create this tubular structure. We understand now the things like hypoxia or ischemia resulting into the hypoxia-inducible factors. They activate these number of relevant molecules creating angiogenesis. So this is the mechanism in a kind of simplified manner I have shown here. There are other methods or observations in the body. One is the sprouting angiogenesis, so the existing vessel. From the existing vessels, like collaterals are formed by sprouting angiogenesis. We have arteriogenesis, so several vessels can become larger, means the artery can become larger, and that's called arteriogenesis. This is predominantly an adaptation of, for example, proximal arteries. And the other one is obviously the neoangiogenesis or vasculogenesis, which starts from the stem cell and creating the vascular network. So obviously when we were presenting this work that brought us to some kind of conclusion, became very popular at that time when many people were working on growth factors and cytokines, and when I showed this data, and it was I mentioned that there could be a natural bypass. We thought that when we do a kind of protocol where the ischemia or slowly occurring ischemia can result in the production of growth factors, and those growth factors would be helpful for the heart to have angiogenesis and opening up the collaterals, and it will be kind of a natural bypass that the heart can heal itself. Obviously, it was a big news that time, and also our information or whatever the knowledge was also based on the VEGF only, but now over the 15 years or two decades, there's a lot of data came out that only single molecule is not enough to create angiogenesis. But we tried all sort of molecules to bring into the heart and the ischemic area to post-MI to create the network of blood vessels, but it was not a great success. Then they started with stem cell therapy, which could be very relevant to stem cell therapy, could be very relevant event, and perhaps that stem cell is, if we bring directly into the infected area, perhaps that will be much better. So work is still underway in different labs in the world every time we hear. Now, based on all this knowledge on blood vessels and understanding the mechanism, we embarked upon the human studies, and we were lucky to collaborate with the cardiothoracic surgeons, those who were working in the lab, in the operation theater to operate the patient. These are the young children or rather babies, those who develop the multiple problems in the heart. So there is a VST, there is a overriding of aorta as well as the pulmonary stenosis. So due to this, because of VST and pulmonary stenosis, the right ventricle becomes dilated, hypertrophied due to pressure and volume overload, and then these hearts will eventually fail if nothing is done. So surgery was done in our center, and we were lucky to obtain some tissue from this outflow tract because the surgeons were working here to alleviate the pulmonary stenosis from this outflow tract. So it's a obligatory tissue removal because you cannot take biopsy from human heart. It was an obligatory tissue removal, and that tissue which was coming out from the outflow tract was really kind of diamond for us to study all the gene expression, DNA marker, identifying the differential expressions from understanding what are the genes. So that's what we embarked upon. And due to these all three anatomical problems of congenital malformations, these heart develop the right ventricle hypertrophy. So the four changes, therefore they are called tetralogy of Fallot. The hearts are called tetralogy of Fallot, means there are four, tetralogy means four, and Fallot was the guy who discovered this disease, was a French clinician. The data which I am sharing on these patients, the young ones, 12 and 12, and then we have took a lot of time to obtain young tissue from the patients, from the young children and also the adults. And these are clinically characterized patients like pulmonary stenosis and right ventricular hypertrophy. But in adults, when they come back, there were a lot of cases which were operated again because they came back after developing the pulmonary stenosis or insufficiency, pulmonary insufficiency. Regurgitation was another trouble, and then they need to be operated again. So what we were doing, we were directly present in the operation theater, one of us. We were able to get that tiny biopsy of 20 to 100 milligrams, sometimes 150. Lucky situation, depending on the outflow tract and the size and all that. The surgeon was, kind enough to work together with us, because that becomes really challenging in many cases, and I think that is the key. If we can convince the surgeons, I think a lot of translational science is possible to do. So we classical experiments starting from expression array, and also a small piece to do the immunohistochemistry and other quantitative tissue remodeling kind of events which we can look under the microscope and quantify the color. We started to, before starting the microarray, expression microarray, what we did, that we wanted to establish that there is a hypertrophy, so that we can at least link some of the genes to this process, or to this congenital anomaly where hypertrophy is involved. This is by classical measuring, by Gomori staining the cell surface and drawing the myocyte, and then calculating the cross-sectional area of the myocytes. As you can see here, there's a lot of work done, many months, and put into a small single table, where we see that the young ones, there is a significant increase in the cell size, as well as in the adults. There was an enormous, huge changes happening in adults because of the dilatation and regurgitation. So, but here you see that there is also a significant difference in the cell size, and we decided to take these patients to do the DNA microarray. So the technology is very robust. You can understand the expression profile of several thousands or hundreds of thousands of genes. You can identify novel genes or functions, dynamics of the genes, and as Kapil mentioned in his presentation, it's really very powerful tool to understand the gene expression profile of a particular tissue, disease tissue, and compare with the normal. We did this using, when we started, the chips were not really so, like now you could do the whole human genome, but we had about 13,000 genes on this chip, starting in a classical fashion, total mRNA to poly A, and then double labeling it with the UTP and CTP, and creating this probe from cDNA probe, and then the cDNA probe was obviously denatured, and these chips were hybridized overnight, and then after washing and doing the staining and the scanning, and we get data something like this. Every tiny dot is a gene representing, and we can really look these dots and convert into a table. So again, a very expensive experiment, hundreds of thousands euros as these expression arrays, the time when we started, but now it's obviously available much cheaper. Even you could do the entire genome sequencing. About many genes which we looked at in the table in the Excel sheet, that these were the genes related to the proliferation that were down-regulated, and many genes which were linked to the extracellular matrix were up-regulated. But interestingly, in the whole list, we found BGF also up-regulated. That was something also a bit puzzling, but we sorted out what were the reason why they are there. So you can see in the differential plot, there are many genes up-regulated, 228, and many genes down-regulated, about 200. We decided to do, in the first row, the fibrosis as well as the vascular component, to quantify, localize all those molecules which were present there, and quantifying those molecules in order to understand these two mechanisms which are directly linked with the hypertrophy and failure. These were established in some other rat models earlier. So we decided to do this first. As you can see, the collagens, the total collagen were significantly higher, ranging from 5 to 7.5. Likewise, many other genes, fibronectin, laminin, others, I'll show you. We can quantify these total collagens by video image analysis, staining the tissue with picrocetus red. This whole red is the picrocetus red or collagen, interstitial as well as perivascular, and we can quantify using the video image analysis, like staining and tissue, and then you can dig out the background. So you get exactly the color, the pixel value of the color versus the tissue. And we can, plot that information in the tabular form or histogram. You see the perivascular, quite a lot collagen already. The young ones, myocytes are much smaller becoming and rather taken over by fibrosis. And as you can see, a perivascular also large amount and as compared to control. When we quantify all these molecules, we see that there is an interstitial collagen as well as the perivascular significantly higher in both failing hearts as compared to the normals. And when we do the differential expression like collagen 1, collagen 3, fibronectin, we see that there are differential expressions. So for example, interstitial collagen 1, when we look at the collagen 3, also interstitial but fibronectin in both the compartments. So the perivascular seems to be more laminin and fibronectin rather than collagen. They are more interstitial. So new information, new understanding coming from this work, which is pure translational work. If you put all these genes, this is just one example. We can look at many crosstalk between the different pathways. As you can see here, if you take these collagens, they are interlinked with several transcription factors, which are again another cluster of tubulins, which are also very relevant for the homeostasis of the cellular system. So if something is changing at collagen level, it will alter many other pathways. So we have to be really, putting things in perspective. It's not only the collagen for these failing hearts, but also because they are linked with the many other pathways, many other cluster of genes which are also changing in response to what is happening there. We looked at the vascular component, as I said, the second one, the VEGF is already about twofold increase, but receptor very little, but is still very relevant because these are very potent margin, and that the receptor is just present, slightly higher, doesn't matter, even if it is present. This twofold increase in the margin can result in the long-term enormous angiogenesis in the heart. It was very puzzling why there should be angiogenesis. If at all there is angiogenesis, then these hearts should not fail, they should survive. But that was not the case, which we established by doing the further experiment I'll show you. There are some more genes which are very relevant, where also TGF-beta was downregulated because TGF-beta is known around the same time that it is a cardiogenic. So like when the myocytes are formed, TGF-beta played an enormous role, and downregulation means that less myocytes will be formed in these patients. So if you look at this, we have the myocardial expression we measured by mRNA and protein receptor here, and also here it's comparable. So the data which we saw in the list on the gene expression, it is confirmed by mRNA studies like happenstance. So there is a significant change in the mRNA level. When we look at the cellular distribution of the protein for VEGF, it is there in the myocytes and but also perivascular or the smooth vessel cells of the blood vessel. but in myocytes it appears like granules in the cytoplasm. So myocytes produce BGF and secretes it out. And then we have done the staining for CD31 to count the blood vessels in these hearts so that we can count every blood vessel. And also we have done the staining for alpha smooth muscle actin. But this is the KDR of FLIP1. It is predominantly present in the vessel, mainly in endothelial cells, as you can see here. So both the receptor and the ligands are present in these patients. How we did in order to establish what is happening to these blood vessels, which are formed because we found large number of blood vessels. We looked at by doing the image analysis by staining them with alpha smooth muscle actin and then measuring this cross-sectional area by the external and internal diameter and calculating the size of the blood vessels, indicating whether this blood vessel is mature or not mature. When the value was less than 3, should be less than 3, but that was not the case in these patients. So the number of vessels were higher in both young one and adult failing hearts, but if you plot them at a capillary level, which were enormous in number in these cases, but they were not significantly altered in size, indicating that these vessels are there just like a mesh, but they are not conducting vessels or not able to withdraw the blood. Hence, as we term these vessels, stunted angiogenesis means these vessels are present, but they are stunted. They are not able to, salvage the myocardium. They are not able to take the blood from the main coronary artery. So, doing this whole expression, cellular, and looking at the vessels in these patients, we can conclude that there is enormous fibrosis in these patients, young ones as well as in the adult ones. We need to find out if there is a possibility to develop a therapeutic strategy, which is not really easy because it's a irreversible process, but perhaps maybe there is a chance that there is a drug which can, bring back the myocytes, because the myocytes will change their phenotype to produce more matrix. So we found enhanced fibrosis. We also found enhanced angiogenesis, but it was stunted angiogenesis. And the clinical relevance of our work is by assessing the appropriate timing. So when, starting from the patient to the bench and then bringing the information back to the bed, to the clinicians, cardiothoracic surgeons, guys, look at if you can operate these patients much earlier than half a year or later when you are doing, because there is a enhanced fibrosis and it's so rapidly changing the myocytes, and these hearts are already in a bad state when you are operating. So now they have decided that they do the surgery in two batches. So as soon as baby is born, they immediately operate to alleviate the BSD. So they put a patch to close the hole in 2 chambers, and then after some years, the patient need to be operated for alleviation of the pulmonary artery stenosis. So that's how the work is done, and these patients are doing very well after. So our information again became very relevant to the surgeons, that they are able to take decision on a case-to-case basis and minimize the damage to the heart so that individual can live longer. So this has been the work which I wanted to share with you, how we start with the experimental work, how we translate that information and directly work on the patients, and how we can generate the data which is so relevant for the catering the matrix therapy or better management of the patients. This way we do the translational research. So I would like to thank you, all of you, and obviously there's a lot of work to be done in this case because we have 427 genes. This is just one example to put them in perspective in a metabolic pathway, linking one of the gene, and you see that the green part showing the upregulation can result in, when you bring into the, like for example, arginine and proline metabolism, which is one of the component and also the matrix. Yeah, there's so much changes. One gene can